Hi, my name is Elliot Richards. I'm a PGY7 at the Cleveland Clinic, and today I will be sharing my research into a novel therapeutic strategy for endometriosis. As I will explain, we discovered that a unique combination of ultra-low dose compounds increases endometriotic progesterone receptor in vitro. This compound may potentially increase the efficacy of conventional progestin therapy. So here's a brief overview of what I'll be talking about. First, we'll talk about progesterone receptor expression in endometriosis, or PR. Then I'll introduce the concept of quality by design, and then I'll share our novel therapeutic strategy and preliminary data that targeting PR in endometriosis. So I don't need to tell this audience about the tremendous burden that endometriosis has on our patients, their families, and their healthcare system. Endometriosis affects an estimated 10% of reproductive age women. It costs an estimated $54 billion a year in the US, and it's a leading cause of chronic pelvic pain and infertility. Endometriosis is defined as the presence of endometrial glands and stroma in ectopic locations. It's most often caused by the retrograde menstruation of menstrual debris, leading to the adhesion, implantation, invasion, angiogenesis, and persistence of lesions. These endometriosis lesions, once established, maintain their own microenvironment of estradiol and often become resistant to progesterone. So altered PR expression is critical to progesterone resistance. This is something that has been shown all throughout the literature, and I'll just briefly share 4 studies here. So in 2000, it was shown that progesterone receptor PR is low or absent in ectopic endometrium of patients with endometriosis, and that when you induce endometriosis in mice who do not spontaneously have endometriosis, you see a progressive reduction in PR over time in these induced lesions. And if you look at patients who have a higher PR expression, their pregnancy rates are higher, and not only that, their response to conventional therapy is better. So patients have lower pain scores if at the time of their surgery, they had higher PR expression. So from that last study, Flores et al. At Yale, this is a figure from their study. And they show, so this dark brown color, that staining there is PR. And so they have 3 groups here with high, low, and essentially absent PR. And they're able to stratify into 2 groups. And those who have high PR show the best response to therapy. So we then have a group of low PR patients who have a poor response to progestin therapy. We have a high PR group that have a high response to progestin therapy. Can we take those low PR patients and convert them to high PR? Can we sensitize their lesions? Is there some type of drug therapy that we can give that can make it so that progestin therapy is more efficacious? And instead of trying to find some new exotic drug, can we take existing known compounds and take them at really low concentrations? Well, this is actually something that our lab has done once before. So this is a paper we published last year. We took a cocktail of low-dose drugs that could get breast cancer cells to upregulate their estrogen receptor, ESR1, in vitro. And we showed that this sensitized them to tamoxifen therapy. And we did this using quality by design. So quality by design is utilized extensively by the manufacturing and pharmaceutical industry. So in quality by design, we begin with a predefined set of attributes of the final product, or the output, and then we identify parameters that must be controlled to achieve this. And one of the key tools in quality by design is something called design of experiments, which I'll talk about. Now, what's really important is that the end goal here, it must be consistently and reliably achieved, even if the underlying mechanisms leading to the desired result are not fully understood. So even if we have a black box, what's important is that we can confidently achieve our output given a set of inputs. So in determining which inputs would be necessary for the desired output, a conventional approach would be to do something called a one factor at a time approach. So in this, to try to find what the optimal result is, we take a given parameter, in this case temperature, test each level, find the optimal level, move on to the next parameter, and before moving on to the next. And this process is very time and labor intensive. And if there are any synergistic interactions between these factors, you might miss the true optimum. Now, using a design of experiments approach, one can reduce the number of experiments needed to find the optimum. And this example is just showing 3 factors, but the more factors studied, the power of DOE becomes really apparent. And it's also important to note in this conventional approach, the true optimum might not be right where it's showing. It might be somewhere else in that design space, but because you've done one factor at a time, you potentially missed it. Whereas design of experiments, you're starting with truly designing out the experiments and really exploring the design space. So it's not only more efficient and faster, you potentially get the right answer. So practically speaking, this means that we take a 96 well of plates, we make individual drug combinations with our compounds of interest, and then we provide these cells and drugs to our collaborator at Trailhead Biosystems. And through a robot at the Trailhead lab, very precise amounts of each drugs are administered to each one of these wells according to a predefined perturbation matrix. RNA is extracted, and then a cDNA library is made. And then we use statistical software to build a model and then select compounds that, when put together, optimize the final outcome based on the results from the cDNA library and expression data. So we use this strategy to target progesterone receptor in endometriosis. So we took 12 candidate compounds. And so these are listed here. We chose these compounds because they were either actively being studied in endometriosis, they were known to influence sex steroid expression, or they were used in the treatment of endometrioid ovarian cancer, which is associated with endometriosis. Now, it's really important to keep in mind that the concentrations shown here that we used in our experiments are well below the IC50. So we wanted to minimize side effects as well as any off-target effects of our treatment therapy. The idea is that we are gently nudging the cells to upregulate the target receptor, which in this case is progesterone receptor. So we have our 12 inputs, we have our desired output, which is progesterone receptor, and we want to optimize for this outcome. And if we were to do this in a one factor at a time approach, this would be well over 4,000 experiments, but we were able to do it in a single 96 well. So here are the results from the model created from our statistical software. So we found 5 compounds that increased PR expression. So siloxib's factor contribution was negligible, so we didn't include it in further study. Paclitaxel had a detrimental effect on some other genes of interest in our cDNA library, and given its role as a chemotherapy drug, we elected to focus on the top 3, which are belinostat, everolimus, and bentamapimod. So these 3 together we dubbed BEB and used for further study. So this plot shows the additive effects of belinostat, everolimus, and bentamapimod on PR expression. And really what the take-home point is here is that maximal expression of these genes is only achieved through combination therapy. So our preliminary work at validating BEB, it confirms in vitro that it does upregulate PR expression, but really importantly, it doesn't affect ESR1 expression. And that's obviously important, as I said from at the very beginning of the talk. Endometriosis is an estrogen-fed disease. We want to increase PR, but we certainly don't want to increase ESR1 expression. So here's a quick summary of our model and our interventional strategy. Basically, PR in this group of patients, their PR is diminished, but by treating with BEB, we can increase progesterone receptor expression and therefore improve response to progestin therapy. And importantly, by using these ultra low doses of these compounds in BEB, we can limit off-target effects and drug side effects. So the next step is really in vivo testing of BEB. And for this, we're going to use our mouse model of endometriosis. It was actually last year at CLESOG that I presented our work of developing this mouse model and how we used it to study endometriosis. And so not only will this provide a means for us to really test a response in PR expression, but we can also get, we can look at lesion size and number in mice treated with BEB. So with that, I'd like to thank my mentor, my collaborators, Trailhead Biosystems, and as well as you for your attention today.